Hello, I'm Maria Williams with the Collaborative on Health and the Environment. CHE enjoys bringing you the latest environmental health science through our partnership calls and webinars, science serves, publications, and social media. I would like to welcome everyone to today's CHE EDC Strategies Partnership Webinar, which is titled, Does Early Bisphenol A Exposure Cause Hyperactivity in Children? A Systematic Review. Our moderator today is Jerry Heindel, Director of the Program at Endocrine Disruption Strategies at Commonweal. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speaker to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speaker. This webinar is scheduled to last for 30 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Jerry.
Okay, thank you. Well, good afternoon, everybody. I'm pleased to introduce our speaker today, Joanna Rochester. Jo received both her MS, and her PhD from the University of California at Davis, studying the effects of endocrine disruptors in birds. But she's currently a senior scientist at the Endocrine Disruption Exchange, also called TEDX, and has worked at TEDX since 2012. So her expertise is in physiology, endocrine disruption, and systematic reviews. And she's currently using systematic review techniques to explore the relationship between chemicals and human health. In the last few years, she's authored several papers on the health effects of BPA, including a review of the human studies on BPA and a review of two alternatives to BPA, BPS and BPF. So she's really one of the biggest experts now on BPA and also on systematic reviews. So today she's going to highlight her expertise in both of these areas, in BPA and systematic reviews, in the discussion of her recent paper, Does Early BPA Exposure Cause Hyperactivity in Children? A Systematic Review. Welcome, Jo, and I turn the virtual podium over to you.
All right, thank you, Jerry, and thanks to CHE for hosting this webinar today. I'm going to be discussing our new study on the link between bisphenol A exposure and hyperactivity. But before I launch into that study, I did want to give a little background on how we came upon this research question. So at TEDx, we are interested in studying health conditions with unknown causes that have been increasing in the past few decades. Many of these diseases or disorders may be caused by exposure to endocrine disrupting chemicals in the womb, which program the fetus for susceptibility to diseases later in life. This is known as the prenatal origins of disease. And this is pretty difficult to study, especially in humans, because you have to test pregnant women for chemical exposures and then follow their children throughout life to see effects. It's a little easier to study in animal models, such as rats and mice. This is because these animals reproduce and mature quickly, and you can dose them with known exposures. But with animals, it can be hard to relate the timing of exposures and the health effects back to humans. So one way of tackling this problem is to review all the published human and animal literature available on a certain disease or endpoint, and then we can start to arrive at some conclusions. One area we are really interested in is neurodevelopment, especially in children, as the causes of many neurological disorders are still unclear. So we chose to focus on attention deficit hyperactivity disorder, or ADHD, for the subject of this review, because it is the most common behavioral disorder in children. ADHD tends to be more common in boys, although this may be due to differences in the symptom manifestation of boys and girls. And these symptoms include hyperactivity, inattention, impulsivity, and deficits in executive function. Recent reports have shown that the number of children who are diagnosed with ADHD has risen to as high as 20% in some population. So that's 1 in 5 kids. And this is compared to 3 to 5% in 1970. There's also significant costs associated with this disorder, up to $52 billion per year. And as a reference, all cancers cost about $87 billion. So although heredity seems to play a part in ADHD, it's been difficult to find specific genes that cause the disease, and it's likely that there's a gene-environment interaction causing the disorder. This means that a person might be predisposed to having a disease due to their genetics, but that environmental exposures might trigger that disease to happen. In order to identify chemicals that might be associated with ADHD, we searched PubMed, which is an online database. One of the problems when doing these systematic type reviews is that there can be a lot of literature to go through. For example, if we just search PubMed for ADHD alone, we come up with tens of thousands of studies to go through. So to pare that down, we formulated other search terms to limit the number of studies. These terms included disease and symptom-specific search terms, as well as prenatal terms, because we were interested in early exposures, human and rodent terms, and environmental exposure terms. This can be a bit of a balancing act because we don't want to have to go through too much literature, but we also don't want to miss any of it or too much of it. And this balance can change depending on what type of review we're doing and the purpose of the review. For example, for this first ADHD search, it was a broad search, and we call this type of review a scoping review or scoping the literature. We're not as interested in identifying every single study, but in identifying as many potential chemical contributors as possible. So this is the results of the scoping part of the review, and this graph shows the number of, the number of studies we found. And we've separated animal studies and human studies, here in green. And we only included studies with positive results. Although I didn't list all the chemicals here, we found over 20 chemicals that were related to ADHD. And this indicates to me that brain development leading to ADHD is really sensitive to chemical exposures. And that's not surprising. It's not surprising to me that ADHD has been increasing in the chemical age that we live in. You can see here that a lot of research has been done in plastic and plasticizers, especially in animals. And this literature is mostly studies of bisphenol A exposure and hyperactivity. Bisphenol A, or BPA, is a well-known endocrine disruptor. It mimics estrogen and interferes with other hormones. As most of you know, it's present in food can linings and packaging, polycarbonate plastic, and on thermal receipt paper. BPA has been shown to be correlated with many health effects in animals and humans. A few years ago, I published a review of the human BPA literature and found effects including reproduction, thyroid disruption, metabolic syndrome, and immune disruption. There were also several studies showing neurodevelopmental changes in children when exposed prenatally to BPA, including ADHD symptoms. So because of this strong literature base and the recent emphasis on BPA research, we decided to perform a more in-depth review of this literature using systematic review. We used a framework developed by the Office of Health Assessment and Translation to carry out the systematic review. OHAT, as it's known, is part of the National Toxicology Program, which is headed by the National Institute of Environmental Health Sciences. The OHAT framework is a 7-step process that integrates different types of studies to come up with a final conclusion to the hazard to human health. And this is known as the hazard identification conclusion. This framework can be carried out with or without meta-analysis, and it does not require a large body of literature to carry it out. It has protocols in place to address study quality and other merits and deficits in the literature. So as I mentioned, this framework uses both animal and human evidence. It can also incorporate mechanistic evidence, such as cell culture studies, but I won't be discussing that in detail in this talk. Animal evidence in general can be robust, and it's often used for regulatory decision-making. However, it may be less accepted by the medical community and the public as relating to human health. As I mentioned before, the human evidence is generally sparser than animal evidence because it's more difficult to produce, and it can be considered less strong because it measures correlation, not direct causation. What's so helpful about this systematic review framework is that it systematically integrates both animal and human literature to come up with a very strong conclusion. OPAC started developing this framework about 5 or 6 years ago, but to our knowledge, we're the first group that has taken it through all 7 steps to reach that final ID conclusion, hazard ID conclusion. So this is our review. It was recently published in Environment International and co-authored by Ashley Bolden and Carol Kwiatkowski. I'll go through each of those 7 steps of the review and the conclusions. Step 1 is problem formulation. Based on our results from the scoping review, we formulated the question, does early BPA exposure cause hyperactivity in humans? For step 2, we searched online databases for studies using BPA and ADHD search terms. We then went through each title and abstract in a process called screening. We used a program called Distiller SR, which allows us to quickly include or exclude studies. So here's just a screenshot of Distiller. And you can see that words can be programmed to be highlighted in different colors. For example, the chemical names are highlighted in blue, the symptom words are in green, and the environmental exposure and human words are in purple. This greatly facilitates screening, and also 2 independent reviewers screen the literature, which decreases human error. Here's a flow diagram of the study selection process. We initially found about 1,500 studies, and you can see that there's several exclusion steps, including full text exclusions. At the end of the screening process, we found 32 studies, which was 29 animal and 3 human studies. Only 12 animal studies were included in our meta-analysis, which I will discuss in a bit. For step 3, we extracted data such as strain and species, sample size, age, and results. Hyperactivity in rodents is measured as increased activity compared to the control animals and can be assessed in many ways. It's a pretty common endpoint looked at in behavioral studies because it is sensitive to perturbation. So researchers will look at the distance a mouse or rat runs around in an open field area, or how many times it crosses lines or light beams that crisscross the area. In a few of the studies we looked at, the researchers put newborn pups on a measuring scale and measured how much that scale moved up and down to assess activity in the pups, which are pretty non-mobile at this stage. For humans, the activity was measured by surveys of teachers and parents of the children. These can be subjective. I think a lot of parents would consider their children to be maybe a bit hyperactive. Sometimes, I know I do. This is a picture of my daughter. So having both teacher and parent input is important, and these surveys have been validated. Step 4 is determining the risk of bias of the individual studies. So don't worry about reading this table. I just included it so you can see how we can look at each study, which is here along the side, and use questions to rate the study. And the questions are here across the top. The risk of bias of the study is the potential of the study results to accurately reflect the true results. So this can be a measure of study quality. This is based on the methodology of the study and the reported methodology. Bias can be introduced in the methods if the study protocol does not follow rigorous standards such as randomization, blinding, and appropriate statistics. So to determine the risk of bias for the studies, we ask a series of questions for each element of the methodology. Depending on the answers, we rate them high or low risk of bias. So in this table, green is a lower risk of bias, which can be considered higher quality, and yellow and red are a higher risk of bias, which is lower quality. The risk of bias results are easier to assess when we group the questions into separate domains. So here we have 7 domains: selection, confounding, performance, etc. And you can see that in general, the human studies had lower risk of bias, or more green, than the animal studies. In a lot of cases, the authors did not report the answers to the questions within the studies, so we attempted to contact them, and we had about a 60% response rate. So if they didn't respond and we couldn't get an answer to the question, we gave a yellow rating for that question. The rodent data did not have a clear outcome when we looked across all studies, so we carried out a meta-analysis. We decided to focus on one dose of 20 micrograms per kilogram per day, as there's the most evidence for this dose, and it's below the EPA accepted safe dose in humans of 50 micrograms per kilogram per day. And there were 12 studies that were included in this analysis. So here's a forest plot of the studies. The females are here at the top, the males at the bottom. The black dots are the outcomes of each study, and the lines are the confidence intervals. If the dots are to the right of the 0 line, we have a positive correlation, and to the left, there's a negative one. And if the confidence interval crosses the 0 line, the effect size for that individual study is not significant. However, with meta-analysis, you can statistically combine each study to come up with a single meta value, and these are the red diamonds. As you can see for males, the diamond lies to the right of the 0 line, which is a positive significant effect. So on the whole, males have increased hyperactivity when exposed to BPA, and we did not see a significance in the females. In step 5, we rate the confidence in the body of evidence for the animal and human literature. So we do this by upgrading or downgrading our confidence based on several criteria. This can be a pretty complicated step, and I'll go over it quickly, but I'll be happy to answer any questions if you want to contact me later about it. So you can see that this step is broken up into 3 parts. The upgrades are represented by green pluses, and the downgrades by red minuses. For step 1, we determine the initial confidence. We start with 4 pluses, 1 plus for each of these aspects of the methodology: controlled exposure, exposure prior to outcome, individual data, and comparison groups used. In step 2, we apply any downgrades. In this case, we downgraded once for high risk of bias of the literature, especially in the areas of randomization and blinding. In step 3, we upgrade for any of these factors. We upgraded once because we saw a linear dose response in the males across all studies. And this makes our final rating 4 pluses or high. For the human studies, we start with 3 pluses because we don't have controlled exposure in these human studies. You can't necessarily dose humans with BPA. So we downgraded, we start with 3 pluses, and then we downgraded once for unexplained inconsistency. And this was because when we examined 3 studies, the significant effects were not consistent across age groups and between boys and girls. We also upgraded for dose response because we did see non-linear dose responses when we looked across all studies. So in this case, our final rating is moderate. For step 6, we convert the confidence in the body of evidence to confidence in the health effect. Because we saw significant positive effects in both the animal and human literature, we have a high rating for animals and a moderate rating for humans. And the last step is to integrate animal and human effects for a final human hazard ID conclusion. A high rating for animals and a moderate rating for humans translates to a presumed health hazard ID. So remember how important this step is. We're taking all the animal evidence and all the human evidence and merging it to make an assessment for human health. So this presumed health hazard is a strong conclusion. So again, the results of our systematic review indicated that BPA is a presumed hazard to humans as a factor contributing to hyperactivity. Other useful information from this review characterized some of the heterogeneity or the variance in the body of evidence, especially between males and females. The review also identified research gaps and revealed more questions. For example, there is limited research on the timing of exposure related to outcomes. Further, the risk of bias for the animals' body of evidence was high, particularly in the areas of randomization, blinding, and statistical analysis. Because it can be difficult to get a good response when contacting authors, future studies need to make sure the risk of bias is low when designing, carrying out, and reporting methodology. So how can conclusions from systematic reviews like this be used? Well, this review would be a great basis for further dose and risk assessments for the purposes of regulation. We did not assess dose other than to assess a dose lower than the established safe level. So this work does need to be done, and this review would be a necessary initial step for that, especially because we did find effects below that safe level. We think that for the public and medical community, systematic reviews can be a guideline for avoiding exposures, and we do recommend that children and pregnant women avoid BPA exposures, which is in line with the recommendations of ACOG. This can be done by avoiding canned food, plastic food packaging, and thermal receipts. Also, it's widely known that brain development is very sensitive to chemical exposures, and our results from the scoping review showed more than 20 chemicals that might be associated with ADHD. So it's worth investigating the mechanisms of ADHD since so many chemicals potentially disrupt the system. If we understand mechanisms better, we could even test new chemicals in cell cultures or in animals before they are put on the market. And lastly, one of the most frustrating things about studying BPA and pushing for further regulation is that there's so much literature, but the regulation is going at a snail's pace. When my human health review came out in 2013, there were about 90 studies total of BPA effects in humans. We've been working on an update to that review, and there have been, since 2013, there have been hundreds more just in the past 5 years. Because they're so researched, we believe these systematic reviews could be the push needed towards faster and more streamlined regulation, and we're currently working on more of them. So with that, I just want to thank our funders and the TEDx staff, especially my co-authors, Ashley and Carol, and to Chris Ribbons, who procures and organizes all of the literature for our reviews. And please check out our website and follow us on Twitter at endo_exchange. And I'll be happy to take any questions.
Okay, thanks a lot. That was a terrific overview. So we do have some questions. So the first question is about the source of funding. Were there instances where it was important to look at source of funding? You said you didn't notice that at all.
You mean like funding of the individual studies?
Yeah.
Any kind of, yeah, so we didn't, I think we did keep track of that, but we didn't consider it in our review. There has been a push to start including funding sources and any kind of conflicts of interest in individual studies as potential bias in those studies. but we didn't do it here. And we did include industry and academic, and I don't know if we had any government. I think there might have been a government or two report in there too, but no, we didn't.
Yeah. So, okay. So what about the mechanistic evidence for BPA and hyperactivity? Can you mention a little bit about that? Where is that?
Sure, that's a great question. So we there's a way to include mechanisms in the actual systematic review to give you a final answer. And we decided not to do that because there's no real strong and accepted mechanism for ADHD at the moment. There's a lot of theories and a lot of research being done. And these tend to look at the dopamine system and serotonin systems. So we did include in the paper a whole discussion of mechanism, and BPA in both in animals and in vitro and cell cultures can disrupt dopamine systems and serotonin systems. Mm-hmm. So there's definitely some involvement there, but we decided not to incorporate it into the actual systematic review.
And in the human studies, did you look at the exposure assessment, especially with BPA? A lot of the human studies only measure it once or twice during pregnancy or early childhood. And it seems to me that's a real risk of bias there, that they don't have a really good exposure assessment. But yes, you didn't point that out for the human studies.
No, so BPA actually leaves your body, I think, in about 6 hours. It's has a very short half-life, I guess you would say. So it is difficult to measure in people and kind of expand that out to how much exposure they're having, say, over a week or over a year or over their whole pregnancy. When you measure a urine sample, it is the urine sample maybe of, it is the exposure from the day before or a half a day before. There have been a few studies that have shown that these spot urine samples, as they're called, do kind of reflect exposure, because if someone's being exposed to BPA, it's on a very, what's the word? It happens regularly. We're exposed all the time. So if someone eats canned food, if someone touches receipts, if someone uses plastic food in their house or plastic food packaging, they're constantly being exposed. So it is hard to look at that with humans, but a spot sample can be a pretty accurate measurement of their general exposures.
Okay. So if you look at those numbers on the plastic cans, mm-hmm. Number 7 is the BPA. So if it's got another number, does that mean it's BPA free?
I think it probably doesn't have bisphenols in it. Other plastics do contain endocrine disrupting chemicals. And so what we like to say is generally try to avoid plastics when you're eating and drinking out of them. Plastic number 7 is actually also encompasses things that don't have BPA in it or bisphenols in it. So it's kind of hard to tell. So in general, we would recommend avoid plastic. Try not to microwave plastic, try not to get it hot, especially in contact with your food.
Okay. So there's a question about, is there any information about the persistence of BPA and its metabolites in the environment?
I don't know that much about environmental bisphenols. I know you can definitely find it in the sewage system or water systems that are tested. I don't know that much about it, but okay. I don't think environmental exposure in that way is a huge concern, contaminant concern.
So I was kind of surprised when you showed the listing of all the chemicals and the little black dots, whether they were positive or negative. There's so much variability, You would think in animal studies they can be controlled better and you wouldn't have so much variability. Is this variability really due to the problems in the assessment of the behavioral endpoints between different studies?
This is just me guessing because we didn't do we didn't look at sources of variability, but I would guess the different strains make a big difference because they're different strains are differently sensitive to BPA and chemicals. And also when the animals were dosed and when the animals were assessed. So were they assessed as adults? Were they assessed as juveniles? How the BPA was given. Was it given as a diet or in their water or injected? So there were a lot of different methods used to look at this, and we didn't account for those because we just didn't have enough studies to look at the differences.
So you mentioned the 20 different chemicals that are involved in ADHD. Is that list somewhere where people can get at it?
We used to have it up online. I don't know if it made it through to our new website, but I'm happy to give that to anyone who's interested.
Okay, so one of the people on the call was interested in that.
Yeah.
And I think maybe the last question. So you've done several of these systematic reviews now. Once you've done a couple, do they really get significantly easier, or is it still really a long, tedious process?
So this is our first full, I would say, systematic review, and we've done other elements of systematic review. It has been getting easier. and I would say to someone who's setting out and starting one, if they're having difficulty, push through, and they do get easier. And there's been tools developed, software that helps people screen. There's some software being developed that will maybe even automatically help you screen studies, prioritize studies to screen so you don't have to look through thousands of them. It's definitely getting easier.
Okay, Maria, I think our time is up. I'll turn it back to you.
Okay, thank you, Jerry. We are approaching the end of today's webinar. A video recording will be available on the CHE website soon, and tomorrow you will receive an email containing a link to the recording. CHE's next webinar from the 20 Pioneers Under 40 in Environmental Health series will take place Thursday, May 31st, and is titled Emerging Issues in Oil and Gas Production, Data Gaps, Policy Challenges, and Novel Threats to Health. You can RSVP through CHE's website, healthandenvironment.org. CHE's next webinar from the CHE EDC Strategies Partnership will take place Wednesday, July 18th, and will feature new research from Dr. Emily Rissman, professor in the Department of Biological Sciences at North Carolina State University. Details will be available on our website soon. If you are new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate these CHE partnership webinars, bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank Joe Rochester for taking the time to talk with us today, and Jerry for his excellent moderation. Thank you so much for joining us, and have a great day.